Thank you, Vicki. Now we're going to move on to our case studies, and Carolyn Compton will start us off. Carolyn is the CEO and president of Critical Path Institute. Carolyn brought her passion for all things standards to CPATH with her from the NCI, where she was director of the Office of Biorepositories and Biospecimen Research.
Thank you, Lynn, and thank you for the organizers for inviting me to this chance to be a case study in data standardization, data pooling for a very specific purpose, which CPATH is engaged in. And it points up, we are, in fact, a good case in point for a lot of the points that have been made by speakers today, including the ones that Vicki just made. What CPATH actually does is act as a trusted third party to develop standards, measurement standards, method standards, and data standards with our 41 industry partners and all of our academic partners. But we do this for a very specific purpose. The purpose is to formally submit these consensus-driven standards to the FDA for a qualification, which is a formal process prescribed by them for pre-review and approval. Once they are approved in this qualification process, they go out into the public domain and stand as a standard for anyone, whether or not you participated in their development. This is exactly how we do it. We are a not-for-profit 501c3 that has been launched by the Critical Path Initiatives program of the FDA. We convene consortia to bring together the best science, and in this fashion create shared risk and shared costs for the creation of these standards. It also drives consensus, and we iteratively include the FDA during this process, so there's regulatory participation and guidance at every step, and then at the end we officially prepare a qualification submission to the FDA, and then they either approve the standard or not. And this is something that they call a drug development tool. One of these standards, they actually have 3 categories of these, which they have defined as biomarkers, clinical outcomes assessment, and animal models, although CPATH does not engage in animal model standards development except where it impacts preclinical studies for toxicology. Now, the FDA, this is a particular slide from one of Charles Cooper's talks from CEDAR, that outlines the need for standards from the point of view of regulatory approval of products. The point being made here is that non-standardized electronic data limits the quality and efficiency of the review process. It's difficult to integrate data sets, it limits the ability to ask in-depth questions, and it increases variability and increases time for review. And therefore, these same issues are affecting the qualification process of the standards that we're developing, since these go to review groups, scientific review groups, for scientific approval. I'm going to pick out one of the 6 global consortia that we have built since 2006 when we began. These consortia do operate globally because all of our company members are global companies. We have more than 1,000 scientists and 41 companies, the who's who of big pharma, participating in this consensus science effort. But I want to pick out one of our consortia, the Coalition Against Major Diseases, which is focused on diseases of the brain and peripheral nervous system, to illustrate a specific use of standards to develop tools for drug development. And basically the point of developing drug development tools and standards is to address the challenge that we now have in this country and internationally of the unsustainable time and costs that it takes to get a new drug to market. More than 10 years and more than $1 billion to get one single drug to market. The focus of these standards is on process improvement for the entire drug development and translational research pipeline. So in CAMD, the tools are to specifically advance effective treatments for Alzheimer's and Parkinson's disease. And we do qualify biomarkers as a drug development tool, but we also develop data standards, create integrated databases of clinical trials data based on standardized data, and built on top of those standardized databases, develop accepted for use quantitative disease models that allow you to model clinical trials, to optimize clinical trial design. And in fact, the very first CDISC therapeutic area, that is disease-specific data standards, were developed for Alzheimer's disease within CAMD and published in September 2011. The start point for us in establishing this is true shared data, coalesced, pooled, shared data, starting with 9 member companies who agreed to share their control trial data from 22 clinical trials in Alzheimer's disease. The data were not in a common format, and they needed to be combined in a consistent manner in order for us to be able to build our drug development tool. And all data were remapped to the CDISC standard and de-identified and then pooled. The result was that we were able to build a new in silico modeling tool that was really based on our ability to create this database. The contributing organizations had to do some work in order to make this happen. They had to go through a corporate approval process to share data and to de-identify it for secondary use. And we further, in CPATH, de-identified the data to conform with HIPAA safe harbor requirements. And this is just one illustration of the heterogeneity of the application of a single so-called standardized cognitive test, the Alzheimer's disease assessment score for cognitive impairment. This is a standard tool for neurologists that are used to assess Alzheimer's disease patients. And you can see here that the implementation of this test across those 22 trials differed in the number of questions, in the order of the questions and tasks addressed, and even the same item was scored differently between trials. So this really required standardization before we could go on to the final phase of building our modeling tool for clinical trials. This required data from 3 different sources. We took data from ADNI, which is a natural history study, where very much like the Framingham study, where the interpatient variability and patient-specific factors, along with imaging and CFS biomarkers, came to us as part of the data set. This was not standardized by a CDISC standard, but all the data was harmonized according to a standard used in one data set, and all of the rest of the data sets that were contributed to ADNI followed suit, but they were not in the same standard that we used to construct our database. And the third source of data was reliant on published data on treatment effect. We could not get companies, even in our own consortia, to contribute treatment level data for our database. We're working on that, and there is some sentiment that we may be able to achieve this. But the point is that we would not have been able to build the modeling tool, the clinical trial simulation and modeling tool, without this placebo effect data coming from our standardized database. It would have been impossible. And what this tool allowed us to do, again, this was a very specific purpose for which we build this standardized database, but it allowed us to merge these data flavors to create a tool that would parse patients into specific groups that would allow you to see differences in disease trajectories over time. These were all 65-year-old males who were not ApoE4 carriers, so they all look alike in the databases, but in fact, when you were able to merge the placebo effect, the natural history, and the treatment effect data, you were able to see that there are three classes of patients, all with different trajectories of disease. And seeing this kind of a distinction emerge from the modeling tool would allow you to design a trial much more wisely. It would inform patient selection, study size, study duration, study feasibility, and even study costs. And in fact, our colleagues at Pfizer have told us that if 4 years ago they had had a tool like this, the bapineuzumab failure that just occurred may have been a completely different trial, or may not have been done at all. So really, when we're talking about the value proposition of data standards, I agree totally with Vicky and with Eric. I've heard him say the same thing, that it's dependent on your research goal, what you want to accomplish. And so the value proposition of building a database like this is really based on the goal of being able to develop a drug development tool that could possibly revolutionize the translational research, at least when it comes to clinical trials, that piece of it, which really is where most of the money, time, and effort is spent, and where most of the spectacular losses are seen. So if we focus our shared data on databases built on standardized data to develop these new drug development tools, we really believe that, and rough calculations that we have made, that we can cut drug development times by 4 to 5 years. And the approach that we use for Alzheimer's disease is now being applied to other research projects. Other organizations, foundations, patient groups have noticed what we've done in Alzheimer's disease, and we are, in fact, doing for them what we did for AD in Parkinson's disease, polycystic kidney disease, and tuberculosis, funded by the Gates Foundation. We did gain new insights in doing this. We learned that legacy data conversion, just like everyone else has said, is resource intensive but worthwhile for specific purposes. All told, the building of that database for Alzheimer's disease, the merger of all of those patients yielded a database of 6,100 Alzheimer's disease patients. And it took us 9 months to de-identify the data and to convert it to a data standard. The assurance that is needed that a specific data set will be useful, it really needs to be assessed up front. Do you need to do this? And then if you decide that you do need to do it, I again agree with Vicky, it's all about selectivity. So use the data that you need, convert the data you need, maybe not everything. And this point was brought up in previous discussion as well. Unless, of course, it costs you no time or no money to do so. New insights can be obtained from data converted to a common standard and aggregated to enable queries and analysis that would not be possible from a non-standardized database. You can't Google a non-standardized database. You may be able to build ways to draw the data out, but either way, you're spending time and resources either to build the conversions or to standardize the database, and it's a balancing act as to which is more convenient. And someone said this earlier, that you always learn something new when you go back and look at the pooled data, and that was exactly our experience. The addition of standardized data from other sources, prospective and or retrospective, actually becomes simplified and expands the power and utility of a standardized data resource once you build it, and if you have implemented that standard in a prospective fashion in other studies. And then your database just continues to grow over time and in power. I want to point out that these kinds of drug development tools that we are developing with our partners in CPATH, and based on the data standards that we are developing with CDISC, are really primarily applicable to these phases of the drug development pipeline where it involves clinical experimentation, but they could also apply in specific instances to post-approval monitoring and gathering of data and insights from standardized data as well. I do want to point out that, as was pointed out earlier, that the Prescription Drug User Fee Act goals actually talk about clinical terminology standards with the goal of completing clinical data terminology and detailed implementation guides for data standardization by 2017. So there still is this focus by the FDA having specific goals for the development and use of data standards, but again, for a very specific use. And from CDER comes this list of 55 therapeutic disease areas that they have put on their website, and for which they are asking the development of therapeutic area data standards within 5 years, the so-called 55 and 5 list. This challenge has been taken up really by the new Transcelerate group. They also recognize the need for data standards, at least for product approval and their submissions, and they're recognizing that the FDA is calling for these to decrease review time. And we are at CFAST now working with Transcelerate as well to develop the data standards for their therapeutic area priorities within the Transcelerate initiative. And this particular session also focuses on governance considerations for data pooling and data standardization. So I just wanted to end with some of the governance considerations that we've had to face in doing what we do. There are actual governance rules for developing the data standards themselves and the need for collaborative expert input and consensus. Rules of the road for merging the data based on our best practices would involve use of high quality data, use of data standards that the FDA accepts if you are focused, in fact, on regulatory approval efficiency, and using data standards end to end. And the rules, there also are rules for accessing the data. We've talked about this. Our database is open to investigators who are qualified. It goes through a data access committee, but at the end of the day, the tool that we're developing on top of that will be completely publicly available. And then rules for access to the qualified drug development tools will be completely free. So that is one good case in point for the data standard use for a specific purpose focused on FDA. Thank you.